recording. Edit. All right. So from today, we're gonna move on to the sort of a big leap actually to the other type of macromolecule that is widely seen in the living organism, which are carbohydrates and then glycoconjugates. All right. So carbohydrates. And they are also called saccharides, especially in chemistry. And I mean, I don't even need to, make a definition for this, right? Everybody knows that whenever you eat something, breads or anything like, beverages or anything, they should contain the carbohydrates, unless you eat something that is pure only, with minerals or so. Whenever you taste something sugary, like sweet or so, those are carbohydrates. So it's pretty much everywhere. We have a slightly different kind of, definition in biochemistry and especially chemistry too, that is a little bit more inclusive. So we're going to cover the properties and definitions and the functions of carbohydrates first. The properties. The basic formula of carbohydrates are basically this, CH2O, and that's going to be a multiple of that. And that N, the multiple of this moiety, has to be at least a 3, meaning that how many carbons do you have to contain in the carbohydrates? It's not a hard question, right? It's basically 3 or more. These carbohydrates are the most abundant class of organic molecules in nature. So, you're gonna see a lot, more than half actually. And the carbohydrates basically contains the little sugars, monosaccharides, as well as disaccharides, oligosaccharides, polysaccharides, and things like that. Depending on the how many of the polymer chains you have, it actually includes all of them. So these carbohydrates, especially for the smaller size, can be a building blocks for polysaccharides, as well as nucleic acids and glycoproteins. And glycoproteins are the proteins that contains the polypeptides with the conjunction to these little sugars like glucose. Nucleic acid, I'm pretty sure we know that the nucleic acids contain the carbohydrates too. What is it? Where is it? What are the 3 components? I'm not. We're just going really back to the sort of the first quarter of these semester. So what are the 3 components, the 3 parts of the nucleic acids? Anyone? Of course, you can forget. That's completely fine, right? Because we're done, basically. That's okay. You got one at least. You on the left. Exactly. There was a sugar, right? Sugar base, which is penose. So we actually have that there, and nitrogenous base as well as the phosphate group. So definitely nucleic acids contain the sugars, and DNA is really bulky, right? A lot of them anywhere in the cells. So that means you're actually having a lot of sugars everywhere. These carbohydrates have many different functions in the biochemical reactions and for sustaining the life too. They serve as a metabolic precursors to virtually all other biomolecules, again containing DNA, RNAs like the nucleic acids, some proteins, as well as lots of the polysaccharides. And they also play a key role in energy metabolism and storage too. So you've heard of glycerols, like cellulose, and those kinds of things, basically forming the other components of the living systems. Are basically comprises the carbohydrates there. And metabolism, of course, we're consuming carbohydrates to get the energy out of that. In the same line, the breakdown of carbohydrates provides energy to sustain life. And it also provides a structural scaffolding for the cell walls, especially in plants. So again, cellulose is one good example. And these carbohydrates are involved in many molecular recognition important for cell growth and transformation, especially these glycoproteins are involved there. Depending on what kind of decorations, sugar decorations those proteins have, they can actually exert the other outcomes for those kind of signaling pathways. And similar thing, glycolipids and glycoproteins play important role in structure and recognition. So they're doing a lot of stuff basically in the living system. And again, like I told you, there are a lot. So more than a half of all organic carbon on Earth is stored in just the two carbohydrates here, either as a starch, which you can see in corns and fruits and breads and everywhere, basically. And the cellulose, again, this works as a scaffold for the forming the cell walls in plants. And when you look at these structures, so this is a simple starch structure, and this is a cellulose structure. They're using the same little components as glucose, but their linkage is slightly different. Actually, the same as like C1 to C4 link, but it has a different conformation that we're going to talk about in this lecture. There are characteristic features, especially in chemistry, for the carbohydrates. This is just a representation of a carbohydrate, especially the glucose. You can see 6 carbons here linked together to form this sugar structure. And these carbohydrates, especially the monomers, the monosaccharides, have one or more chiral centers. And we talked about chirality, basically. What is being chiral? What is chirality? Can anyone explain that? Yes? I didn't catch that. Exactly. So the chirality is basically derived from the a compound that is centered by a carbon usually, which has the 4 valency, and it is attached or making the covalent bond with all different functional groups in each valence, basically. So it has that tetrahedral structure, but it's does not gonna have how do you say, the symmetry there. So it can create the mirror image, and the mirror image cannot be overlapped, perfectly overlapped with the between those mirror images. That's the chirality. So that means again, since these carbohydrates contain carbons, and these carbons, as you can see, have a maximum valency, attaching to the different functional groups, it can create the chirality there. And these carbohydrates, which looks a linear form here, can also make the ring structure too. And again, we're going to talk about that process too. And it can also have the polymeric structures like oligosaccharides or the polysaccharides by the making glycosidic bonds. And this is not about just making a sort of a linear, the chain, something similar to the amino acids, but it can also branch. So it can make all the branches like using the hydroxyl groups on the sides too. As you can see, there are a lot of hydroxyls here. And there are going to be extensive amount of hydrogen bonding that are going to be involved for these molecular interactions. And the reason why is because you have a lot of hydrogens as well as hydroxyls, right? And each hydroxyls can basically create the hydrogen bonding with other polar molecules like water. So it can be fairly well dissolved in water, like sugars, as you know, can easily just dissolve into water, right? They have, when you look at the classification, especially for their sizes, they're categorized as monosaccharides, which are the simple sugars, just a single sugar molecules. Disaccharides with the one molecule with the 2 simple sugars, And oligosaccharides are basically like a tens of those simple sugars are linked together. And polysaccharides, it's like really extensive form like a starch or cellulose. And they also have all different kinds of isomers. And we talked about the isomers before, right? But especially stereoisomers are applied here where they have the same kind of arrangement of the functional groups, but will have a different orientation. So that's what stereoisomers are about. And there are subtypes of stereoisomers, which are epimers and anomers, that are really useful, to tell which this sugar is, the identity as well as their chemical properties too. Again, we're going to talk about this in more depth in short. Okay, so like I said, monosaccharides, which is a simple sugar, is really important because it basically forms the property of the polymer too. These monosaccharides are modified polyhydroxyl alcohols. So as you can see, this has hydroxyl groups, lots of them, so polyhydroxyl. And alcohols, when you look at it, in the end, they have hydroxyl group, and that is directly attached to carbon. It makes this kind of alcohol structure here. And these monosaccharides, like glucose or fructose, can be classified based on the number of carbons from 3 carbons, which is triose, tetrose, 4 carbon member, sugar basically, to the heptose with the 7 carbons. And also can be classified by the nature of carbonyl group. And carbonyl group is right here within the aldehyde functional group, which is a CHO. And whether you have the aldehyde or the ketone group here, which is a CO. Again, we talked about this long time ago, in the beginning of this semester. So hopefully, if you don't, recognize this too well, then go back and, sort of have a refresher with that. For and the based on the what kind of functional groups you have the sugar attached, like the carbonyl group or the ketone group, you can call them as aldose for the sugars containing carbonyl group, the aldehyde, and the ketose for the ones that containing the ketone groups. And for aldoses, if it contains 3 or more carbons, they can have the chiral centers. As you can see, I mean, if it has just one, you don't have chirality there, it's just a CHO. And 2 of them still cannot do that because this carbon will not have another, R group there. But once you have 3 of them, then this carbon in the center can now have HOH, aldehyde, and the other R group. So it can be a chiral center. Question?
Are they like classified based on like the number of carbons and the carbonyl group, or is it like either or?
Either or. So they can be, classified in many different ways. And these are the 2 examples, basically, that are widely used for their classification. So, of course, you can just simply say penose, right? Which is used in the nucleotides, as opposed to you can say that this is aldose, which is containing the aldehyde group. Depending on what you're focusing on, what kind of things you want to say, you can actually classify them in the different ways. Doesn't mean that they're mutually exclusive, right?
So is there like one way that's like more preferred, or is it just whichever?
Yeah, whichever. I mean, basically, nothing's really. I mean, all these are pretty common, so you can choose anything basically when you're just calling them. That's fine. But basically, if you want to focus on the carbon groups, you can use these classification as opposed to if you want to focus on just the number of carbons. For example, talking about the chirality or the ring structure and things like that, you definitely want to use these terms, right? Hexose and so on. Any other questions? Yes. For the aldoses, right? Are you asking about aldoses? Okay, about the where are you talking about? This one? So I didn't get to that yet, but we'll definitely talk about that. So, all right, and that is kind of cute that I need to go a little faster, right? So no, I'm just kidding. Anyway, so for aldoses, whenever you have three or more carbons, you're going to have a chiral center. As opposed to for ketoses, you have to have one more carbons, four or more carbons, because one of the carbons are attached to this oxygen with a double bond, right? So it'll never be a chiral center. Can't have that tetrahedral structure. So you have to just put this aside and have to think about other carbons. Hopefully it makes sense. If not, just ask me now. And we talked about the chirality here, so I'm going to just skip it. In glucose, now we're getting there. 4 out of 6 carbons are chiral, and the question was that which carbons were the chiral centers? What do you think? Anyone? What are the 4s? Yes. So there are numbers. Carbon numbers are there, so make it a little easier, right? So I was gonna do this a little later, but let's just define the carbon numbers first. So you always start with the aldehyde when you're numbering the carbons. So it always starts with this top one as C1 or carbon number 1, and then C2, C3, C4, C5, and C6, basically for hexoses. And of course, I mean, you can have the trios, tetros, and things like that, but same thing applies. So you always start with this aldehyde group or the carbonyl group, and then just numbering them. So now we're all set. Yeah. 2, 3, 4, 5. How come? Exactly, that's the right answer. So when you just look at them, just put that carbon in the center and think about all the functional groups that are attached. If they're all different, then they are chiral centers, basically. And you can apply to that. The second one, H, OH, aldehyde, and something else, right? Same thing applies to this middle one, OH, H, some kind of functional group, the other type of functional group, right? As long as they're not identical, these two R groups are not identical, they are still served as the chiral centers. So what about this cis6? What is it? Exactly. It has 2 hydrogens, so it's going to be symmetric. So it's not going to create the chirality there. And the similar thing can be applied to the ketoses too. Yes. No, yeah. That's right. That's a good question. Where should we start numbering the carbons for the ketoses? We could have started from this ketone because it's ketosis, right? But the problem is that now you have to go back to this one and just count it as cis6, so it's a little inconvenient, right? So instead of doing that, we always just start it from the carbon that is closer to the ketone group. Make sense? So that's going to be 1 to 6 in this way. And where are the chiral centers in this hexose ketose? Anyone? Number 1? Yes or no? No. Number 2? No. Number 3? Yes. Number 4? Yes. Number 5? Yes. Number 6? No. So that's why aldose is with 3 or more, and the ketose is with 4 or more carbons are going to be chiral centers. And then you can create a lot of stereoisomers basically, because you can, it's whether how you arrange the hydroxyl group, right? You put it on the right hand side or left hand side, right? So that's going to be 2 different kinds of combinations exist. And basically for the 4 chiral centers, you can go up to 16 possible combinations for the stereoisomers. So there are a lot. And since this is a little bit complicated structure, even if they have a really simple functional groups, still those are 6 rings, 6 carbons, right? So it's a little complicated just to draw it in a proper way, in a correct way. So instead of doing that, we use Fisher projection here too. And hope you remember how we actually use the Fisher projection, where we have this molecule, especially we use the this glyceraldehyde as a sort of a standard, right? And then these horizontal lines in the Fisher projection here, which connects the H and O between the carbon, is going to be basically projecting towards you, as opposed to the vertical line, meaning that they're retracted. They're projecting against, like away from you. And you can apply for this Fisher projection scheme to the glucose or the other monosaccharides or the what is it, carbohydrates, basically. But when you take into account, so the important thing is about the location of this hydroxyl group, right? And when you look at this sugar, it has the same CH2OH moiety here and CHO moiety on the top. So it's easy to just put it that way. But now we have questioned whether which hydroxyl we need to use for telling that whether this is D form or L form. What we use is the farthest carbon from the carbonyl group. And we're going to talk about the regions later on, but keep in mind that we're always using the farthest one. So in this case, but still you have to take into consideration whether this is making the chirality or not, right? So that's going to be, instead of C6, C5, right, which is the last chiral center. And you look at the location or the arrangement of this hydroxyl group, which is on your right-hand side. So that's going to be D form. If it were on the left side, then it should have been called L-glucose. And that farthest carbon is the same as the highest number, for the asymmetric carbon, again, chiral centers. And D form sugars are predominantly seen in nature, not because this form is more stable. I mean, you can have D forms and L forms. We talked about it when we talk about enantiomers, basically. But and also we talked about whether it is optically active or not for these mirror image enantiomers, basically, which have the mirror images of the same components of the molecule. They're going to have the same property, exactly the same property, same propensity for dissolution or whatever that is, reactivity with the other molecules and stuff like that. It's exactly the same. So you could have actually used the randomly D forms and L forms of the same molecule. But the nature actually chose to use D forms, not because D form is more stable, or more advantageous, but because that was how the evolution just chose. You can just refining the enzymes and that are making, having the reactions with those molecules, basically. And if you're going to use both forms, it has to be more like evolved, right? Have to have more energies and have to take more considerations through the evolution, as opposed to if you choose just the one form, you can be specialized and more optimized easily with one form. So just happen to be a lot of them, exist in the D forms. And we talked about the enantiomers here, D forms, L forms. This is not about the enantiomers that we're telling D or L forms. It's all about the last chiral center, right? Whether the hydroxyl group is located on the right side for D or left side for L. Any questions so far? Okay. And I briefly talked about epimers and anomers, right? So we're just keep covering about these kind of definitions of the different isomers here, because these are really important to identify different simple sugar structures. So epimers are the basically among the stereoisomers, but it's a pair of isomers, stereoisomers, that are not enantiomers, especially for sugars. And that is the same as diastereoisomers, right? You can have stereoisomers, and they can be either enantiomers or diastereoisomers. And that's depending on whether this one has the mirror images or not. Make sense? So, but these epimers have only one stereogenic centers, or the chiral center, that is different in their configuration. Meaning that let's take an example here, comparing the D-mannose and D-glucose. When you look at all the functional groups, they're the same. The arrangements are the same except for this C2, where it has OH on the left-hand side for the mannose, but the right side for the glucose. So these two, mannose and glucose, are epimers because they have only one different configuration that is not enantiomer, that is not mirror image, right? And this happens at C2, so they are called C2 epimers. Another example here is D-glucose and D-galactose, where you can see that everything is the same except for the C4 arrangement for the hydroxyl. So in which case it is called C4 epimers. Make sense? What about the D-mannose and D-galactose? Are they epimers? Are they? Why not? C2? Mm-hmm. C2 and C4? That's correct, because they have different configuration for two carbons, right, at the same time. So they are not epimers. For epimers, there has to be only one stereogenic centers that have different configuration. Make sense? So you got it right. All right, so these are the different types of aldoses and their structures, basically. Starts with the trias, have the 3 carbons, and 4 carbons, tetroses, and 5 carbons and 6 carbons. And these are particularly important, ones that are boxed out are really important. As you know, D-glucose, D-mannose, D-galactose, these are really widely used as sugars, simple sugars. So you should remember their structures, basically. And when you look at this, the ribose, and you should be fairly familiar with this ribose because it's a component of RNA, right? And DNA too. If it has the loses one hydroxyl, and let's compare this, the ribose and the arabinose. When you look at the other, so of course both of them have aldehyde and the acetyl group on the bottom, and of course they have all the hydroxyl group attached to all the carbons there. But it's all about the configuration, right? The second one, when you look at the C2, it has the different arrangement for the hydroxyl group. The rest of them are the same. So are they enantiomers? No. Are they epimers? Yes. So they are epimers. Same thing applies to D-glucose and D-mannose. They're epimers, but C2 epimers. And again, glucose and galactose, when you look at it, the same except for the C4. So they are the C4 epimers. Okay? Similar thing is applied to the structures for and the types of the ketosis here too. It starts with the this dihydroxyacetone, which has the ketone right in the middle, and then you can basically add the this HCOH moiety one at a time to make it as the bigger molecules. And you can see that some of them are basically, epimers like that. And an interesting thing is that you can get these all different kinds of ketoses in nature as a monomers, monosaccharides, as opposed to the only aldose as a monosaccharide that you can get is glucose. And this is because they use a different pathway for the proteins, not the protein, sorry, molecule synthesis. Ketoses are synthesized basically inside your body using the different smaller chemicals or the big, smaller smaller molecules or the bigger molecules, as opposed to glucose is basically obtained by the intaking or the uptaking the bigger molecules, and you just chop it down through the metabolism. So you're gonna only get that glucose or the fructose, basically, all the simple sugars for the aldoses group. All right, so let's delve into more chemical properties of the sugars. So we so far talked about the monosaccharides, right? These are the same as a simple sugar, just a one molecule of sugar. And when you look at the sugar, the most reactive group is carbonyl group, again, aldehyde, where it has a CHO moiety right here. And that basically tells how the cyclic form is formed and how polymer is formed. The polymers and the cyclic forms and all other modifications happens around or on this carbonyl group because it's the most reactive. And the reason why it is reactive is because of this oxygen and the double bonding to the carbon, which has lots of electrons there. So it can basically attack the less electronegative atoms like carbons of the other molecules, and can make the bonding or get rid of hydrogen and so on basically. So this aldehyde can react with alcohol to form the hemiacetal. And for ketones too, which we can see in ketosis, can react to alcohol to form hemiketal here. And these are the structures. And this is the basically formation or cyclization of the sugar from the linear form. And as you can see, the sugar itself contains this acetyl group. Within that, there is a alcohol, right? Right here on the bottom. And here's a CHO, which is basically aldehyde. So there's a really reactive functional group here, and here's a alcohol. So you can actually make this reaction happens even within this sugar molecule. And that's the driving force for making this cyclization, circular, cyclical form of the sugar. And similar thing applies to the this ketosis too. Let's see. So I already talked about this, and again, you should at least understand how this cyclization happens. What kind of functional groups are involved for this cyclization? And there are basically two different types of cyclic, forms of sugars. Pyranoses are derivatives of pyran, and there's a structure of a pyran here, which have a six-membered ring. So it has 1, 2, 3, 4, 5 carbons and one oxygen molecule. And another one is a furanoses, which are the derivatives of furans. So they have a 5-member ring. Okay. And for pyranoses, which is a 6-member rings, are the really important and widely used form for the simple sugars like glucose. So when you look at the glucose, again, this is the structure of a D-glucose, has the aldehyde group on the top, and the alcohol on the bottom within this acetyl group. And these guys can react to each other. Here, when you look at it, here's alcohol, here's aldehyde, and they make this hemiacetal structure. And when you look at this structure, which is about to be, be a cyclic form, here's an alcohol, OH, and here's an what is it, aldehyde group here, and then basically this looks exactly same as this, right? C here, and the OH here's just the O with the double bonding, but will become the OH, getting protonized, and here's the R group, here's an H. So it's exactly the same, structure here. So it forms the hemiacetal structure, and then will be eventually be in the cyclic form. And depending on where you put this O, top or bottom, it actually makes in this different configuration. Can be, the hydroxyl can be on the bottom of this six-member ring, or them on the top two. And based on that, we can actually call them as a different isomers too, that we're going to talk about briefly, in short. And this, when you look at this ring form, the C1 right here, which forms the aldehyde, is now become chiral. Can you see that? So it is used to be not chiral, or doesn't count it as isn't counted as the chiral center when it was in the linear form. But once it makes in this ring form, now it has O here, H here, OH on the bottom, and the R group here, right? So basically, this becomes chiral center now, and it's called anomer. And anomer is a type of stereoisomers, and but it's different from what is it? Sorry, I'm blinking a little bit now. Epimer. So we talked about the epimer, right? So it's anomer is a type of epimer, but it's specialized for this ring structure, especially the carbon that used not be the chiral center becomes a chiral center when it forms the cyclic form, is called anomers, or anomeric center, or anomeric carbon. And we're going to talk about this again, during this lecture. So don't worry about it if you don't understand what I'm talking about. And furanose is another type of, the sugar ring, but it has the five member there. And an example here is the fructose. And again, here's the reaction to form the hemiketone. Just want to check the time. Here's the alcohol again, and here's a ketone instead of aldehyde. And again, there's a oxygen that reacts with the carbon in this ketone functional group to form this hemiketal. And you can see the same structure in the linearized, not the linear, sorry, cyclic form of the ketones, ketosis basically. So this is a linear form. And when it makes this circularization, you can see that here's a R2, which is acetyl group, and R1 becomes in this rest of the carbons here, and here's a OH that used to be the ketone, but now it'll become the hydroxyl, and another one is the basically the alcohol, the RO, right? So it's the same structure here. But keep in mind that this is different from pyranose, where it forms the six-member ring, right? So all the carbons, except for the last acetyl group, are going to be within the ring. So only this CH2OH is going to be exocyclic, right? As opposed to furanose, since the folding region happens at C2, now this C1 is going to be excluded from the ring. So instead of a six-member ring, it's going to form the five-member ring, and you're going to have two exocyclic carbons. Make sense? So that's the main difference. But again, in the end, you're going to have hydroxyl group from this ketone, and that can be arranged on the bottom of the ring or the top of the ring, and based on their, so again, this used to be not the chiral center, but now it becomes a chiral center within the ring. So it's going to be called as anomeric center or the anomeric carbon, and it'll form the anomers based on where you put this hydroxyl. Question. C2, you mean? Is chiral center. So when you look at this linear form, there's second carbon, C2. Is it chiral center or not? It is not. Once it's circularized, now is it? Is it chiral center or not? It is, right? So now it becomes a chiral center only inside the sugar ring. And that's the definition of anomer again, And the anomeric carbon is basically that particular carbon that now becomes the chiral center. All right? And again, same thing applies for this previous pyranols too, right? This C1 used to be not chiral center, becomes a chiral center. And that's all about that anomeric carbon or the anomers. All right, so now let's talk about the anomers. We kind of going a little backward, but the anomeric carbons are basically the carbons within the sugar that becomes the chiral center, right? And anomers are those sugar rings that contain anomeric centers and have different configuration for hydroxyl group, either going up or down, or when you make this the Fischer projection, whether this carbon, the hydroxyl goes right hand side or the left. So this is a little like confusing. So let's start with the this Howard projection here that we're going to talk about too, about that type of projection. But anyway, when you look at this anomeric center, and it again used to be C1, right? Used to be C1, and if this hydroxyl is on the bottom and the opposite side from the exocyclic carbon, then it's going to be called alpha form. So this is alpha anomer, as opposed to if they're on the same side and I'm talking about exocyclic, acyl group or the carbon and the hydroxyl group attached to anomeric center or anomeric carbon, then they're going to be called beta form. So alpha and beta, which are for anomers, are different from D and L forms, which are stereoisomers for enantiomers, right? This is about enantiomers or anomers. So you can have, alpha, meaning that your hydroxyl is on the opposite side from the exocyclic carbon, and D form, meaning that when you look at the latest, the largest carbon number, which is here, whether this is located on this hydroxyl is located on the right side or the left side. Does that make sense? And you can, of course, have this intermediate stage, which is a linear form, and can go back and forth, basically, going to this alpha anomer or beta anomer. Okay? This one or? You don't understand the third point? Oh, so the C6 is right here, right? Which is exocyclic carbon. Is it? Which is right here. And we just look at the orientation of this exocyclic carbon and the hydroxyl group. If they're on the same side, it's beta form. If on the opposite side, from the sugar ring, then it's the alpha form. Yes, I believe I said that, right? Then they're gonna be beta form. Alpha beta is basically, I mean, you can change this orientation of the exocyclic portion, but you can change the orientation of a hydroxyl group by going back to the linear form and to the other anomeric form. Yes. Yeah, that's a good question. So what about the anomers for? Hopefully, I have that. I don't think so. Um, it always you always take into account of the anomeric carbon. So you just have to look at the hydroxyl groups that is attached directly to the anomeric group, which is a C2 for the ketosis. But for this one, for aldosis, that's going to be C1, right? So just to look at that, focus on that C2 or C2 for ketosis or C1 for aldosis. And whether, since you're going to have the two exocyclic rings, now you have to choose one, right? But one that is far from that anomeric center will be the one that is considered for telling that whether this is alpha or beta form. Okay. I know it's a lot of definitions basically, but what will be really important to go further, further understand this like sugars, their structures and functions later on. So we kept talking about the anomers. So hopefully we covered and we're on the same page in terms of what the anomers are and how we can tell that whether this is alpha or beta forms, how we can identify the anomeric carbons and so on. And anomers can interconvert between alpha and beta forms spontaneously via it always have to through this linear intermediate. It will never just rotate and shift in this hydroxyl up and bottom because it's pretty stable there. It has to use a lot of energies to turn it basically because it's a bulky region, bulky group, right? So you're gonna have experienced a lot of steric clashes if you wanna just turn it over. So instead of doing that, you just break the link and go back to the linear form and then go to the other anomeric form, basically alpha to beta. And again, this is a spontaneous process. So, and this process is called mutal rotation, where there's an interconversion between the different anomeric forms. And an example is that even if you start with the pure one form, like a beta form, you're going to end up having a mixture of some of alpha forms and some of beta forms. And this is an example of glucose, actually, which I forgot to write down here. And the concentration of the linear form, which is an intermediate, is really teeny. How come? I mean, in most often cases, you're gonna have either alpha anomeric form or beta anomeric form, but you're gonna have only little chance to have this linear form. Why? Can anyone make a speculation? What's that? Oh, my question was that can you guys hear me well on the back? Okay. My question was that how come you have so little of this linear form, but predominantly have only the cyclic form, alpha or beta? Because here, I'm saying that the linear form, the concentration of linear form is small. Yes. That is absolutely true, but it could be, that's true. So energy, stability, it's kind of similar thing, right? it definitely can be. But how come? Why does the cyclic forms have lower energies? Why is it more stable? Everybody's connected. But when you look at the linear forms, they're all connected too. I mean, there's nothing floating, like around there. No electrons or anything that is floating there. Nothing's really kind of, there's a clear aldehyde group here, clear, alcohol here, which is acetyl group. And all of them are hydroxyls here, um, basically filling the max valency there. Exactly, you got it. It is because of this not the hydrox, the what is it? The aldehyde, or the acetyl group that is really reactive. So it try to actually react with something. That's why. Even if it is a relatively stable form, it still try to just whenever it meets something, some kind of things, it try to just make a reaction there, because that's more favorable. So that's why it'll have, and of course, it's conveniently have another, its partner, which is alcohol on the other end, right? So if you have a longer, uh, this what is it, the monosaccharide, you're going to have a more chance to be, circularized, as opposed to the shorter forms like trios and things like that will have a hard time to make the cyclic form, because it's simply really hard to fold it up, and also have a lower opportunity where these two functional groups are meeting each other. Okay. All right. So I just talked about the projection here, which is called the Haworth projection. But basically, you can use either Fisher projection, but Fisher projection is not ideal for the cyclic form, because you have to basically write it this way, which is a lot like not so like the what the actual molecule look like, right? So instead of using Fisher projection, you can use this more convenient form, Howard projection, where you simply draw this sixth ring here, and this bottom portion, meaning that they are closer to you, and the upper portion is away from you, and you can also easily indicate that whether such certain functional group is on the bottom of the plane, the sugar ring, or the top of the sugar ring, right? And again, depending on where you see your hydroxyl attached to the anomeric carbon, you can tell that this is whether alpha, which is the opposite side, or the beta, which is on the same side for anomers. All right. And when you look at the sugar ring conformation, this is not just a simple flat, but they exist in the either chair form or the boat form here. And this is because of this electron densities. It has the sp3 hybridization on each carbon members, right? As you can imagine, they have a tetrahedral structure. So they're going to have sp3 hybridization everywhere for their electron densities. So to minimize the steric clashes, instead of keeping it as a sort of a flat ring, it'll have this kind of tilted ring with the angle of 109.5 degrees. And all the substituents that are like sort of other functional groups that are attached to those carbons can be located like alongside, along this plane of the sugar ring, which is equatorially, which is perpendicular to the axis here that you can see goes through the sugar ring, like penetrating the sugar ring, or it can also orient it axially, which is parallel to this axis. So that's going to be on the top or the bottom of the sugar ring, basically. And it can have any kinds of forms, basically, based on the shape or the size of those substituents. But there are some rules here, two rules here. The first ones, bulky substituents, the big ones, are more stable in equatorial conformation because it has a smaller chance of steric clashes. And you can imagine that, right? Because there's an angle here. So if everything's, if the bulky stuff is like axially attached, and then they have more chance to just meet each other, get closer, and kind of create clashes. And the second rule is that the chair is slightly more stable than the boat form. And you can guess, that why I'm going to ask, whether I'm going to ask this or not. But why? Why is the chair form is more stable than the boat form? Yes. What is it? Oh, because it has a larger surface area. Surface area is an interesting term, I think. I mean, which I never actually used for the molecular structure, but I totally get that, what it means. Any other person? Yes. Exactly, less steric clashes. So that's correct, and that is actually the same as what you just said. I think it's a creative way to say it actually. So, kind of like that. So that is true. In the both form, there's more chance when you look at these two A's, will get closer as opposed to these two A's in the chair form, right? They're on the completely opposite side. So they're going to create the less internal energy, basically. And the property, let's quickly talk about the properties of these sugars. So chemistry of the monosaccharides or the simple sugars or the chemical reactions, basically, is mostly due to the reactions of hydroxyls and anomeric carbons. We already talked about this, right? The hydroxyls are everywhere here, including the one that on the alcohol too. And aromatic carbons are basically ketones or the aldehyde. So they're highly reactive already. And this anomeric carbons or the carbonyl can react with alcohols to create the hemiacetal and hemiketyls, which are important for making the circularization of the sugar molecules. And they can also create this glycosidic bond between two simple sugars or monosaccharides to connect them each other to make the polymers. Like here in this case, and they're making, here's a one sugar, and it has another acetone here that is attached to that on this region on the around the aromatic center, and later on this can be react to the same molecule to make the linkage through the glycosidic bond. In the end, it'll leave just the oxygen there as a bridge. And we're pretty familiar with this glycosidic bond because we've seen it for the linkage between the nitrogenous base and the pentose rings in the nucleotides already, right? And it's called N-glycosidic bonds because this sugar was attached between the anomeric carbon and the nitrogen on the nitrogenous base. So that's N-glycosidic bonds, as opposed to for sugars, they're going to be called as O-glycosidic bonds. And again, it's the linkage between the anomeric carbon and the O, basically the oxygen molecule. Yes.
In this slide, there was a reaction between an alpha and a beta compound. Do you have an alpha-alpha? Would that be numerically?
You definitely can. So, I mean, basically alpha and alpha, alpha and beta, beta and alpha, that actually determines a different disaccharides or the oligosaccharides or even polysaccharides. And that actually confers the different properties of those polymers, sugar polymers. Like, we at the beginning we talked about starch versus cellulose have the same components as glucose, but it's, if you remember, that was either alpha link 1,4 linkage or the beta 1,4 linkage. It's the same linkage, but whether it is flipped over or the same kind of orientation for the linkage, which is O-glycosidic bond. Here we go. So I guess you just you could have said that by looking at the next slide, but here you can make this kind of zigzag shape for the even disaccharides, but also applies to the polysaccharides too, or this kind of more like a linkage directly on with the same orientation too. And based on those, especially for the one that is attached to the anomeric carbon, is important to tell whether this is alpha or beta form. This is beta because when you look at this, oxygen, which was from the hydroxyl, is same orientation as the exocyclic carbon, right? So that's beta form. That's why it's called beta. And it's an example of lactose. And when you look at these molecules, this is literally galactose. And here's a beta form. There's a linkage from between carbon 1, C1, and C4 in glucose, right? So beta 1, 4, and glucose. And that's lactose. Same thing applies to all of the other disaccharides. Sucrose is another example here. Again, glucose, fructose, alpha, because OH is the opposite side from this exocyclic carbon, or the functional group, and then you have a fructose. And the linkage is between 1 and 2, C2. All right. And well, lactose is commonly found in dairy products, and the sucrose is commonly used as table sugar. And also, we have maltose and many other things that we can actually use many times in many different kinds of, edible stuff. And for the naming, we just talked about this, right? The naming of disaccharides would be, they can have their own name, but basically you can just write it as using the IUPAC, looking at the individual components of that. That's going to be monosaccharides involved and the linkage type. So for sucrose and lactose, we already talked about this. For sucrose, it's going to be glucose, and then here's a alpha anomer, right? And then linkage between 1 and 2 carbons, and then fructose. So that's going to be glucose, alpha, 1, 2, fructose. Make sense? Lactose, can anyone try? It's going to start with the galactose. Exactly. You got it. So this is how you name it, basically. Yes. Doesn't have to be. It can be. Absolutely. That, in which case, just a second, how would you call it? So this has happened to be the linkage between C1 and C2, but the alpha anomeric and the beta form here. But we still call it alpha, right? What if it used actually beta, alpha form here too for the fructose? How would you call it? Isn't it supposed to be alpha beta 1, 2, or alpha 1 beta 2? Yes? Was it a, oh no? Yes? Mm-hmm.
Because like from going to the second glucose, or I guess the only glucose, isn't that an alpha bond? So you could be on that. Yes.
You definitely can. It's all about anomeric carbon, actually. So it counts. As you said, anomeric carbon is located here, right? So that's why. It's all about the anomers, but you don't care about this because it's not an anomer. Can anyone answer to this? 5, right? Can you see that? Go back to that cyclization process. Oh, it's 2 actually. In this case, it's kind of hard to tell actually, because you don't know where it was oriented from the original one. So it's hard to tell. When you look at it, here and here are exactly the same, right? It's just the sort of symmetrical. So it's hard to tell where it was originated. So the origination is important in this case to tell that which one is anomeric or not. But this one's hard actually because you don't know. Exactly. It really doesn't matter actually, right? Whether 2 or 5, there's no clear anomeric carbons here. So it's hard. Besides, I mean, this is much more clear. I mean, it's always anomeric carbon for this C1 in glucose. But in certain cases where you use 2, so in here, this C1 is anomeric carbon, but C4 is not, right? So of course you can't have alpha or beta form. So that's why you skip it here too. But there are cases where you use alpha 1, beta 2, and so on, if you have a clear anomeric carbons there. Make sense? All right. And this is actually the last slide. And for the sugar polymers, you can have reducing end and non-reducing end. And reducing end is basically the end of your polymer of the sugars. So where you have this hydroxyl group. And that means this is reactive to certain things, right? So you can actually open this ring again with the involvement of water or some other molecules and can linearize it. As opposed to non-reducing end is again the end of the sugar, molecule, but that is located within the polymer. So it cannot actually, since it does not have hydroxyl group, instead of it has the glycosidic bonding there, right? So it cannot be opened up and revert back to the linear form. So it's not going to be able to be reduced. So that's going to be non-reducing end. And this is a pretty useful one where you define whether certain chemical, sugar chemical is reduced, that whether you can reduce it or not, reduce it back to the linear form or not. And that actually tells you the identity of the sugars too, like especially for the disaccharides or the oligosaccharides. Yes. Reducing end is oxidized, can be oxidized. Is that what you're asking?
Yeah, so it's not by reducing end, by the reducing end, you mean that the carbon is oxidized to form the 3 double bond.
So the question was that whether this carbon is oxidized or not to be reduced. Is that? So it takes out the proton, right? So hydrogen, meaning that this is deprotonated, and that means it's reduced, or the basically loses the proton, so it's working as an acid. Is that what you're? So it's not going to be oxidized, it's going to be reduced. It's the opposite term when you look at the carbon, because it's losing proton. I'm not sure it's, we talked about the, acid-base reaction by the Lowry, definition, right? So it's gonna be reduced. That's why it's the reducing end. And the reducing is losing the proton. Hopefully it makes sense. All right, that's it.